Hello, my name is Kateřina Faltejsková, and I come from the Institute of Organic Chemistry and Biochemistry of the Czech Academy of Sciences in Prague, Czech Republic. I would like to present to you our new piece of software called Paperfly, the partial assembly-based peak finder. Specific recognition of a DNA locus by a transcription factor can be influenced by the binding site sequence, which has around 15 base pairs. However, multiple experimental studies indicate that larger context of the transcription factor binding motif affects the recognition process, maybe via local DNA shape. Cooperation with other transcription factors is also important in the recognition process. We have intended to see the edges of the larger context around the motif. By means of genome assembly algorithms, we have developed a tool for binding site reconstruction without any prior knowledge, including the reference genome. We take the sequencing reads from a ChIP-seq experiment or a similar experiment that produces reads with differential enrichments. We identify the k-mers in the sequencing reads. We construct a de Bruijn graph, which is a graph of overlaps. Then we assemble the sequences using a novel algorithm that uses pathfinding and the different enrichments of the k-mers seen in the experiment. We align these sequences and estimate their number of occurrences. And then, using probabilistic methods, we identify the peaks in them. We have tested our programs on 300 ChIP-seq experiments from the ENCODE on human transcription factors with read lengths from 70 to 100 base pairs. We have reconstructed peaks with Paperfly and then mapped them to their closest image in the ENCODE. We measured our error as relative edit distance of the reconstructed binding site to the ENCODE binding site. The results can be seen in this plot. On the y-axis, you can see the relative error, while on the x-axis, you can see each experiment. As you can see, our median error never exceeds 3%. However, we still have some room for improvement as some experiments produce high error rates. In this plot, you can see the number of occurrences of a Paperfly peak in the ENCODE peaks. So we can divide the Paperfly peaks into generally two groups. One that has tens or hundreds of occurrences, and second, which has only units, small or up to 10 of occurrences. These two groups are almost partial in size. We are still not sure what causes this phenomena because the sequences in the groups with hundreds of occurrences have little to no similarities in sequence nor in local DNA shape. Therefore, we are trying to test the affinity of these sequences, which have around 10 to 150 base pairs, experimentally, and therefore maybe realize something about specific recognition. So in conclusion, we have developed a software which is capable of reconstructing unique binding sites seen in a ChIP-seq experiment with low error while operating without a reference genome. However, we are not dependent on the ChIP-seq workflow, and our algorithm can be used on any sequencing data with differential enrichments. While testing our approaches on human transcription factors, we have seen that the binding site we have identified can be separated into two groups based on their abundance in the genome. First group can be seen in hundreds of copies, while the second in units. We are not sure what causes this phenomena, and we intend to have our findings experimentally tested. Paperfly can be found freely on GitHub. Here, you can use the QR code to send to you there. Thank you for taking your time to listening to my poster.